All right, it is officially 1 o'clock and we are ready to get started. Thanks everyone for joining us today. I'm Megan Weber, an aquatic invasive species extension educator at the University of Minnesota. And I'm excited that you're joining us as we welcome Alex Bajcz here to chat about some cool modeling work he and a group of collaborators have been doing to help predict Starry Stonewort infestations. I'm going to just have a couple of housekeeping items, and then I'm just going to turn it right over to Alex. So we are going to use a tool called Slido today to help with questions and answers. You can drop your questions in there at any point, but we're going to wait on Q&A until after Alex is done speaking. But we should have, roughly a half hour for questions. So lots of time to go through those. You can access that by going to slido.com and entering the event code STARRY. We'll also, when Q&A starts, have a little QR code that you can scan with like a phone or a tablet if you wish as well. Um, and let's see, if you have any troubles with that, we do have Michelle here who will be able to pull anything that you put into chat and put it over into Slido so that we don't lose it in the process. We will be recording today's webinar, so you can watch for that recording to hit our YouTube channel. It usually takes a week or so for us to get it transcriptioned, but you can view this in any of our past webinars there as well. And everyone will be muted today. So if you do have questions, we encourage you to use that Slido feature to get your questions over to us. And that's about all the time I'm going to take up with housekeeping so that we have as much time as possible to hear from Alex and get some Q&A in. So Alex, I'll let you introduce yourself and take over the screen sharing to get started.
All right. Hello, everybody. My name is Alex Bajcz, and I am the quantitative ecologist for MACERC. But before I was the quantitative ecologist for MACERC, I was actually a participant on a MACERC project, and that's what I'm going to be talking about here today. This is going to be work that I led that finished up late last year, along with all of the colleagues that are shown up here on the slides, including Jeffrey Dozer, who I'll point out because he was the one who actually wrote the R package SP occupancy that we used for this work. I've entitled my talk today, Fanatic Weeds and Where to Find Them, because this work, as Megan noted, was specifically aimed at informing how we should be looking for Starry Stonewort, which is a stealthy aquatic plant invader, so we can find it earlier more often. And I guess I want to start my talk in an unconventional way. I just want to spill all my beans right off the bat because I know I'm talking about a modeling project and these can get confusing. So I've put everything on the slide here that I want you to take away from my talk today. First and foremost, some of you who are observant may have noticed I've spoken recently for the seminar series. I actually spoke back in January. And at that point, I was speaking as MACERC's quantitative ecologist. I was showcasing PI Charter, which is our app for aggregating these PI or point intercept aquatic plant survey data from across the state. I said at that time, those data are valuable, and you'll have to take my word for it. Today, I'm speaking as a member of this MACERC project team that is showcasing one very cool way we are using those data that we are aggregating in PI Charter. So I hope to show you the value of those data today. Second, my talk is going to be centering the unique and varied challenges that are faced by AIS monitors. For those of you who are out here in the room today, you and your challenges are seen and heard. I want that message to come across. That's why we want to help you do your work. Third, though I will highlight a few surprises along the way too, I'm going to be showing mostly good news, and that is that Starry is not doing anything occupancy-wise that we might not have necessarily predicted coming in, which means that our results do suggest that the ways we are currently monitoring for Starry now seem sound and defensible, but they also show that the modeling or the monitoring could be improved if we inform it using occupancy models like the ones that we have built. These are not tools that are used in this space very often, but we show it's possible and valuable to build them anyway. And lastly, of course, we want to acknowledge that, no model should be treated as just truth without validation on the ground, right? Models need to be used to see if they're working, and they're better when they're iterative, right? When we gather data specifically designed to challenge them. And so that work obviously should come next. And that's really it. If you got a busy day ahead of you today, I guess you could switch off now because hopefully you've gotten the gist of what I want to communicate. But if you are eager to dive deeper, stay tuned because that is exactly what we will do. To begin, as I just sort of alluded to, our project is really fueled by these PI or point intercept aquatic plant survey data. And for those of you who caught my January webinar, I apologize. This is going to be a review. But a PI survey, very simply, is the systematic cataloging of a lake's flora. And this is oversimplifying a bit, but in a PI survey, what you do is you cover a lake with a grid, and you take all those intersection points, and you figure out which ones are too deep or too shallow to sample, and you ignore those. All the others you go visit via boat, and you take a metal rake head, and you get it overboard that boat somehow, and you reel it in. And everything that ends up stuck to that rake head is then ID'd and scored for abundance. So here is an example map from PI Charter, actually, that is showing you rake abundance data for coontail, so Ceratophyllum demersum, for a 2023 survey that was done at Silver Lake here in Minnesota. These dark colored circles, as implied by that legend down here, are places where we did not see coontail at all, whereas these bright colored spots are places where the coontail was very abundant on the rake when it was retrieved. And there would be a comparable map for every other taxon that was observed at the same lake at any of these points. So hopefully what you can see here is that this map is demonstrating that every PI survey is this data treasure trove. For tens or even hundreds of spots in a lake, we might know who is there, how abundant they are, and maybe what else is going on, because sometimes other data like depth and substrate data are gathered along with the plant data. And it's really for that reason that MACERT committed with my hiring to becoming this sort of clearinghouse for Minnesota's PI survey data. In January, I regaled you all about PI Charter, this app that we've built that allows everyone to view PI survey data from around the state and to importantly send us more of it. And at that time, I showed you a records tab that was very much still under construction. I have since very much enhanced it. It now has interactive graphs and maps of all the data from every survey that we have. So even if you checked it out in January, it's worth going back just to see what I've done on the records tab, because that map I just showed you comes straight from there. All this work to aggregate PI survey data, though, I don't think would be easily justifiable if these data weren't valuable. So the fact that I'm here today talking about them again hopefully shows you that MACERT views them as valuable, and I hope you'll see how exactly they are valuable through my talk today. All right, with that, I'm going to switch gears to talking about AIS monitoring and early detection, which is really the thrust of today's presentation. As many of you already know, this is the process of hunting for a new AIS after it's established someplace new, but ideally before it's done too much new damage there. And the idea here is, control or eradication is going to be more successful when you discover invasives early. And that's good logic. It's sound logic. It's the correct logic. There is just one small problem with it, though, as logic, and that is that early detection of AIS is bloody hard. Why? Well, first, some lakes are big. In fact, a lot of lakes are big, and I mean really big. Not just two-dimensionally big, but three-dimensionally big, right? You're searching through a three-dimensional space for something. Meanwhile, a lot of the invasives we're looking for are small, right? This picture I just put up here is of a piece of a starry stonewort plant, and it would fit comfortably on someone's fingers, let alone in the palms of their hands. So imagine you're looking for something that small in a lake the size of the picture I showed, right? Couple that with the fact that there's just not enough monitors out there, and it means we can't even approach looking everywhere at all times. Even the places we look, though, right? A rake head is a great tool for surveying plants, but it's area-wise tiny in the scheme of even a small lake. Plus, even at a small lake, if you survey at 100 points, you still may only have sampled something like 1% or less of that lake's area. And then even with perfect technique, a small spatch of Starry Stonewort or something like that could easily be missed by a rake sample. So you add this all up and you get a simple truth, I think, and that is that AIS monitoring is this task where we are trying to find something when it is at its absolute hardest to find. It's rare, it's small, it's hidden, it's maybe not even well understood yet. So it's not even just that it's hard, it's paradoxically hard, right? Combating all those headwinds necessitates knowing which spots are likeliest to have the invasive we're looking for. That means we need to identify patterns about where a species tends to be or not be that are reliable. And to find such patterns, we need data, but not just any data. So to tell you what I mean, or to show you what I mean, I want you to imagine an abstracted lake here, right? It's a single lake. It's infested by Starry Stonewort, SSW, in 3 small isolated spots like this. Now I want you to imagine that I said we're going to hunt for patterns in where Starry tends to be based on a small data set, maybe just 3 data from 3 random spots per lake. As this abstraction hopefully shows you, that's just not going to be enough data, right? If our invasive is rare, a small data set is going to miss too often to teach us anything, right? If you're thinking about Battleship, there's just not going to be enough hits to go off of to feel really good about any of the patterns we're going to find. We can't use a small data set. Even though we're talking about a rare species here, we still can't deal with a small data set. So now I want you to imagine that the data set we're going to use is a lot bigger, but it mostly contains data from easy to reach locations. Maybe, for example, we only have shoreline data because then we don't need to get a boat out to get these data, right? Well, if Starry doesn't exclusively live along shorelines, which it doesn't in our abstract lake here, this data set's going to give us a falsely narrow impression of where Starry tends to be. And a lot of occupied locations are going to go unmonitored as a result, right? We're not getting the whole picture here. We can't use only what we'll call convenience data, right? Similarly, we can't focus only on data that are driven by our preconceived notions of what we already think Starry likes, right? If there's any bias, intentional or unintentional in our data set, it's gonna at best only seem to confirm what we think we already know. It's not gonna necessarily tell us the truth. If we're only looking where we think Starry is gonna be, those are the only places we're gonna find Starry, but that's not likely because those are the only places Starry actually lives, which means a lot of occupied spots are going to go unmonitored, right? We have to use data that at least tries to challenge our assumptions a bit. We also can't rely too much on data that could be fishy or iffy, right? If a species is rare, as many invasives are at first, we can't have a bunch of non-detections making it seem like that species can't live somewhere that it actually can and does live. So we have to be able to trust our absences, our zeros in our data set. Similarly, we can't have a data set that's full of misidentifications. And in the case of Starry, and I know some will push back on this, but it is possible to mix up Starry with other members of the Characeae, especially when they're all growing together and Starry's in the minority and the plants are small and yada, 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 right? We need to be able to trust the absences. So we need these data to be really reliable to trust the patterns that we find. Otherwise, the zeros are going to really muddy things up a lot. Lastly, if we're trying to figure out where a species prefers to be, we can't only include data from lakes or spots in lakes where that species could totally live and might actually even like, but it just hasn't gotten there yet, right? Yeah, it might be absent from all these spots over here, but those absences are about a different process. They're about opportunity, not preference. And so if you have 2 different sources of absences that are really different, you need to keep that in mind. And we really need ones that are more about preference than about opportunity. So that's a lot of requirements. So to recap, we need lots of high quality data from a rare species that are not biased by a location in a lake or convenience or preconceived notions or plant community characteristics. And we need them specifically from places we know the invasive could be living right now because it's had the opportunity to get there. And that brings us to the data set that we amassed for this project. It consists of data from 23 starry infested lakes in Minnesota and Wisconsin, totaling almost 23,000 observations from over 8,000 within lake sampling points over an 8-year period. Many of these lakes were sampled multiple times, and 3 were actually surveyed multiple times in the same years on 1 or more occasions. So this data set really sort of meets head on all those earlier stipulations I just laid out. Biggest thing is it's systematic, right? PI data indiscriminately assess the whole lake, not just spots we had hunches about or were easy to reach or whatever. Second, these are rigorously collected data. These PI surveyors are veterans, they're pros, they do this day in and day out. Non-detections and mis-IDs are unlikely to be a significant issue in this data set. Even if they were, though, there's repetition here, right? We've gone back to the same spots multiple times in many cases. So if Starry's really there, we've had a chance to successfully ID it and detect it in many of those locations. And these were all lakes we knew were infested when these surveys were done. Starry is there. There's no question of whether or not it could get to these lakes or not. There's no opportunity question. So hopefully this shows you why PA data are such amazing data for something like this, right? It should be stressed, though. They were only available because so many partners in both states collected them in the first place and then shared them with us when asked. And we should all be very grateful for that. And I know my team here certainly is. And I'll be thanking the partners involved here again at the end of my talk. So those were the data and why we needed the data that we needed. What did we do with the data? Well, first we cleaned and combined them, right? These are our 23 sets of surveys from a lot of different agencies. So this was actually no small feat. It took me several weeks at that time to do this work, which is why I'm partly so excited that PI Charter can now largely do a lot of that work automatically in a fraction of the time it took me at the time that I was working on this. Second, we added to them, right? We took the plant data that was contained in there and the depth data and so forth, and we added some additional data to these. One of them is fetch. Fetch is basically how far can wind blow over a lake and through a point, because as it does that, it mixes up the water. And some plants like mixing and some plants don't. So fetch is about wind-driven mixing. We also added a bunch of measurements related to each point's proximity to public and private boat launches, because we think boats is how Starry is spreading around. So this will speak to the spread and opportunity processes that I was talking about a little bit earlier. And then we fed them into a special kind of predictive model called an occupancy model, using this SP occupancy package that our collaborator Jeff Dozer wrote. And so the next thing I want to talk about is I want to briefly explain what an occupancy model is and what it does. But I'm not going to do that in math. I'm going to do that in words. So if you panicked when I said that, please don't run away from the screen. Like most models, our model is taking data in and trying to find patterns in those data. So one of the most important sets of data we're putting in here is Starry detection, non-detection data from PI surveys. Where has Starry been seen and when? So these are just ones for yeses, zeros for nos. It's the yellow, the green and red pegs from a game of Battleship, if you want to think about it that way. We also have to put into our model predictor variable data, though. These are our things like fetch and depth and boat launch data. These are what the model is going to be using to hunt for patterns. What do occupied sites tend to look like? What conditions make a site more likely or less likely to be occupied? That right now is so far so model. But occupancy models are not just any kind of model. They have some bells and whistles, and I want to highlight two of these. First, the model knows that zeros, these non-detections in our data set, actually come in two flavors. One of them is Starry's not there because this is not a good spot, right? That's about preference or biology. Versus Starry's not there, or it looks like it wasn't there because actually it was there and you just missed it when you went looking for it, right? You failed to detect it. The model knows that there's two different processes producing these zeros. So not all the zeros are real, right? Those secondary set of zeros, those are actually fakes. Those are actually ones that we just don't know are ones. So as it is going in here hunting for patterns, it's actually trying to figure that out and is actually guessing behind the scene. Actually, you know what? I bet Starry really is here. And I'm going to consider that when I'm looking for patterns. So that's pretty cool. The other thing that's cool is that it can account for spread explicitly. So we all learned with COVID that infestations spread locally, right? Via close proximity. The model knows that occupied sites are going to do the same. They're going to cluster in space. And they're not going to do that because all these spots are necessarily fantastic for Starry. They're going to do this because this is how things spread, right? One of these things got infested and the infested spread locally. The model can actually take that into account and model it. I'll talk a little bit more later about how it does that. So my point here is that we weren't just blessed with fantastic data. We actually also had a really powerful tool to put them into. And that's noteworthy because occupancy models like this are not often used for invasive species monitoring or hunting. And it's not because they aren't fantastic tools for that purpose. They are, but because they demand a lot of fantastic data to put into them. And that's not something we always have, or at least it wasn't until we started aggregating all these PI data. And now maybe the landscape has really shifted here, at least when we're talking about aquatic plants. So that's one of the major benefits to all these PI data that we're gathering up here. So without further ado, I'm going to spend the rest of my talk showing you what the model has been telling us. And to do that, I'm going to show you a bunch of maps. So I want us to practice reading one of these maps first, because I know they're kind of busy. On the x-axis, we have longitude. Y-axis, we have latitude. The shoreline of the lake is marked in this black outline here. And if there are any boat launches, they're marked with yellow diamonds. This one's got a B on it for public, so this means that this is a launch run by some government agency. If you at any point see a V, that's for private. That means it's run by some sort of business or other private entity. Beyond all that, there's 3 forms of information on every map I'm going to show you, and they're going to be contained in 3 ways. The first of these is shape. So if we know Starry is at a spot because we've seen it there, that gets a square. So all these red squares here are places we know Starry is, or at least was, at least at one time. If we're not as sure if Starry is there because we haven't seen it yet, those get circles. And you can see that the circles all have colors associated with them. Those are the model's predicted occupancy probabilities. How likely is it Starry actually is there? And if you see a warm color, that means a relatively high probability. If you see a cool color down here in the navy blue range, that means a low probability. And you'll notice the points have different sizes to them too. That just corresponds to less or more of something. And here that's uncertainty. If you have a small point, that's low uncertainty. The model is pretty confident about its prediction there for probability. If you see high points, that's higher levels of uncertainty. And there could be many reasons for that. So small is always less of something, large is always more of something. On future slides, oftentimes that will be some sort of predictor variable, like maybe I'll make size have to do with depth instead. And so bigger points will be deeper or something like that. But we'll cross that bridge when we come to it. If not all that sank in, no worries. We'll get more practice over the next 10-ish minutes, and we can absolutely dissect these further in the Q&A if you'd like. Okay, let's start talking in the most important place, biology preference, right? What factors seem to correlate really strongly with a spot's starry occupancy? And what we found here is that both depth and fetch correlate here. And that's actually not news. This has been found before. Previous studies have shown, for example, that greater depth and greater fetch are bad for starry, quote unquote. That is actually not exactly what we found. And we can visualize the results we found here using what's called a tile plot first. So a tile plot is just a table filled with colors. And in this particular table, as we go from left to right, we're going from shallow spots in a lake to deep spots in a lake, although still in the littoral zone, so places where plants can still grow. And as we go top down here, we're going from spots with a really low fetch, so low levels of mixing, to spots with a high fetch, so high levels of mixing. Then you can see that there are colors and numbers here, and those correspond with the model's predicted likelihood that Starry would occupy a spot like that one. So if you see a warm color here, high likelihood Starry would like a spot like that, cool color, low likelihood. What we are seeing here in this table is that there's some nuance to this relationship between depth and fetch and occupancy. As we move left to right in this table, as we move to deeper spots, what we are always going to observe is a cooling in color. What this is telling us is that in general, greater depth is generally worse than shallower depth for starry. However, as we do that left to right movement in low mixing areas, the drop off is pretty gradual. It's pretty steep at high levels of mixing. So it seems like, depth is never a good thing for starry, but maybe it's tolerable if mixing's not going to be so high, but it's really intolerable if mixing is high, right? You get really low occupancy probabilities in those cases. With fetch, what we see is as we move from the top down, we're moving from low fetch to high fetch. That actually seems to be pretty darn good for starry if we're talking about a shallow location, but terrible for starry if we're talking about a high depth location. So what we would call this is an interactive effect, and we're the first study that we know of so far that's documented such an effect like this. It's not like previous studies haven't looked at fetch and depth, but they haven't interacted the two in this way. And so maybe they just didn't get to see this relationship before. I promised you maps, so let's look at exactly what I just showed you in map form instead. This is Long Lake in Wisconsin. Recall on these maps, warm colors are a high occupancy chance. Big means more of something. And here, like I said, that's depth. So what you are seeing is there are a lot of large points here. Those are deep spots. They're in the center of the lake, not surprisingly. And they have cool colors pretty much uniformly, which corresponds to a low occupancy probability. That corresponds to what I just said on the previous slide, which is that high depth, not generally good for starry. Most of your worm colors are right along shore here in some capacity or another. Although, even the deeper spots in this lake aren't profoundly deep, I should note. On this map now, I've instead switched us to fetch being related to point size. So you have a high fetch of a large point. And what you can again see is a lot of the large points here are along the center of the lake, but they're specifically along this axis, right? Because you can imagine wind starting here in the southwest and blowing to the northeast, and it would blow over that lake in a long, uninterrupted stretch and maybe mix up those waters extra hard as a result of that. And again, you see generally cool colors along all these large points, and that's because in general, especially if it's deep, high fetch is not so good for starry. But we can start to see some nuance if we zoom in on specific points here, right? These 2 points, both in the deep part of the lake, they have slightly different fetches though. This one is a higher fetch than this one does, and it's hard to see here, but this one actually has an even darker color of blue than this one does. And that corresponds to what I said on the last slide, which is at deep depths, more fetch, bad for starry. We see the exactly opposite thing up here. Both of these are in shallow waters. This point here is a larger fetch than this point here. And this time the larger point gets the warmer color because at shallow waters, high fetch is good for starry or seems to be correlated with starry occupancy. If you're thinking at this point, wow, Alex, that is a really subtle pattern. Yeah, you're right. This is why modeling is really awesome, right? There's dozens of patterns operating in nature at any time. What a model can do is see patterns like this that are lying beneath all these other patterns that our eyes would really struggle to see. So maybe this is why it just hadn't been noticed before that there was some interactive effect between these two variables like this. All right, next I want to talk about axes because we've long assumed that starry is moved around the landscape by boats, so boat launches should be among the likeliest places to find starry by. And that is what we found. As predicted, proximity to a launch was a strong predictor of occupancy. And we could see this here at Little Cedar Lake. So on this map here, what we can see is that nearly all the occupied sites in this lake, as well as all the warmish colors, are all clustered down here near this one boat launch on this lake. But I'll point out, not all the occupied locations at this lake are down by the axis, right? There's some way up here too. This is why I was saying PI data are so awesome, right? Because they're systematic. We'd have missed these if we were just surveying more opportunistically around axes, right? Anyway, yes, our results support this current practice that we have, right? With time being and resources being tight, we're just going to go survey around launches for starry and see if we can find it there. Our results suggest that that is a good approach. But even here, there's some nuance to the story that I think is interesting. First, we also included in the model whether having more launches versus less launches in an area mattered, like if there's an additive effect. We also tested whether type of launch, public versus private, mattered. And in both cases, the answer was no, not significantly, which is pretty interesting, especially that last one, right? This suggests that private launches and public launches are about equally risky. So it seems like maybe we should be surveying near private launches because that would be just as useful as surveying near public launches. And that's not something we generally speaking do now for a lot of reasons. I think this also begs some additional questions like, does that mean private launches really aren't as risky as some people thought? Are interventions at public launches maybe not working as well as we thought? And I think we can't read into these results nearly as much as we might want to, right? Not so fast. The 23 lakes we had actually didn't include all that many with private accesses, especially not high densities of private accesses. So I suspect we didn't have quite enough replication here or representation from private accesses to really get reliable patterns to emerge here, right? I think we can definitely say, being near an axis matters. But beyond that, I think more work might be needed to really draw more conclusions. And I think that kind of work should be done. Anyway, the fact that Starry likes to be near launches of some kind or any kind is actually not new. It's been shown before, but our model may have actually clarified about something about why it tends to be found near axes. Because if you think about it, there's actually 3 ways to explain Starry's apparent love of being around launches, right? Maybe these are just spots that Starry can get to easily thanks to boats, right? Maybe it's about opportunity. But maybe it's actually that these are just genuinely great spots for Starry. Maybe something about these spots makes them uniquely hospitable, right? It's about passion. Or maybe it's somewhere in between the two. Maybe both explanations have merit, right? Well, here our model actually found another interactive effect that I think is very interesting because it favors one of these explanations over the others. So I'm going to put up another tile plot. This time, as we move rightward here, we're moving away from the nearest boat launch at a lake. So as we do that, what we can generally see is a cooling down. It seems like it's generally better, quote unquote, to be by an axis if you're Starry. However, this changes over time. As we move down the table here, we're moving from very soon after an infestation has started at a lake to many years after an infestation has been going on at that lake. And what you see is that the cooling really gradualizes as we do that, right? It seems like early on, you're definitely going to mostly find infestations around axes or pretty close to them. But after an infestation has been going for a while, that really levels out to almost being just as equally likely that you'll find Starry away from an axis. And that is just not what you'd expect to see if areas near launches are uniquely good in some ways. It is, however, exactly what you would expect to see if launch areas are fine, sorta, maybe, but they're stepping stones. They're where infestations start, but Starry basically can't wait to spread out away from them over time. And that's newsworthy because there might be good news contained in there, right? That might mean Starry infestations tend to start in spots that actually aren't all that great for Starry, which means if we can find these infestations early, control might be likelier to actually work because they're trying to get these things out of spots they actually don't love in the first place. I don't know, maybe there's a silver lining there, maybe not. Those are really the biggest highlights. I do want to, however, quickly show you two more things because I think it highlights how the quality of the data we had available helped us build a higher quality model. First, I mentioned earlier, right, spread's local. Occupied sites are going to be more likely to be near other occupied sites, not because all those spots are great, but because that's just how spread works. I said earlier this is a problem because if your model ignores this, it's going to see spread patterns and interpret them as preference patterns instead. So it's going to think some spots are much better than they really are because, wow, there's a lot of occupied spots here, but it's really just because that's where spread was happening. Conversely, it's going to underestimate occurrence around sites you already know are occupied, even though those are near sick spots and they're likely to get sick too. So SP occupancy is a pretty incredible package. And one reason for this is you can include in your models a variable called W. This is basically a weighted average saying, the occupancy probability of a spot should be linked to the occupancy probabilities to its 15 nearest neighbors. If you're sick, your neighborhood's likelihood of being sick goes up too. If your neighbors are sick, your likelihood of getting sick goes up too, right? So there's some stickiness in the model based on spread that in the occupancy probabilities it's coming up with. And you can actually see this play out at Pine Lake. I want us to zoom in here on the center of the lake because this is a spot that should be bad. It's deep, has high fetch, it's far away from axes, relatively speaking, and yet some of the warmest colors in this whole lake are out here. Why? They're by some occupied sites by chance, it seems like maybe. And so the model is saying, these are near sick sites. They should be more likelier to be sick as well. So this is cool because it means the model isn't misinterpreting or ignoring spread, which is the options you might get in a more basic model. It's also cool though, because the math of getting something like this to work is really scary. So the fact that Jeff just built it to work like this is incredible. The other thing I want to show you has to do with non-detections, right? Earlier I said even pros miss things sometimes. Rake tosses aren't perfect, neither are rake throwers. Sometimes you're going to miss Starry even when it's there. This is something the model doesn't just get to think about. It actually models it explicitly. It asks, based on any repeat samples that we have, are we likelier to miss Starry even though it's there in certain kinds of spots? And what the model actually tells us is that overall, on average, it thinks you got about a 63% chance to detect Starry on an average rake throw in an average spot. What strikes me about that is that is not bad at all, but it's far from perfect. Like that's a 1/3 chance of missing Starry in a spot it really is in if you only look once. And the model actually says that actually goes up to more like a 2/3 chance if you're tossing in a spot that's got a lot of plant color cover, especially if those plants are related to starry. So maybe you've got a better chance of starry, blending in or something like that, right? So not only is modeling it like this, making the model make better informed predictions because it can factor that in when it's looking for patterns. But I think even these results themselves are actionable, right? They tell us that the odds of success under different conditions are going to be different. And maybe we need to adjust our approach under those conditions if that's something that we're concerned about. So with that, I'm going to return to the slide, recap the things that I hope you'll take away from my talk today. Yes, without a doubt, PI survey data are a critical resource. I hope you're getting that from my talk here today. But they're not just critical for invasive species work. Aquatic plants in general are understudied and not well understood. I think these data could be a really powerful agent for change in that arena too. Yes, without a doubt, AIS monitoring is hard. We know this. That's why we want to do it strategically and try to figure out how to do it more strategically. And we think models like this could really help with that. And in part, they can help by just supporting or rejecting or countering the ways we monitor now, right? Starry surveillance tends to focus now on launch areas, and that seems like exactly the right approach. However, maybe we need to start doing two rake tosses if a spot is really planty, or maybe we need to start thinking about surveying in high depth but low fetch areas, which you might find in bays. Or maybe we need to start adding private launches to our lists where and if that's possible, right? Overall, though, our model broadly matched our expectations, which is reassuring. It means it passed the sniff test, but it also taught us some new things too, which I think is also very valuable. Now we just need to go out and see if it works, right? Validate it on the ground, see if we can improve on it in some way. And I would hope that's going to be the next step here. All right, to all of those who contributed data to this project, thank you so much. It simply would not have been possible without you. And I hope that you will continue to support our work into the future as well. And of course, thanks to all the funders and thanks to all of you out there for listening to our talk today. I'd be happy to take any questions that you guys have.
Wonderful. Thanks, Alex. All right, I'm just going to take over screen sharing from you real quick. To remind folks on how Q&A works, we're using a tool called Slido. You can either scan the QR code you see on the screen now, and it should take you direct to that event, or you can type in slido.com into your web address bar and enter the event code STARRY. Even if you don't plan on asking a question, you can go in and upvote other people's questions. So if there's ones that you really want to see get asked, you can, tip the scales in your favor that way. There are already some questions here, and I believe we also have one of Alex's collaborators, Dan, as well, to help chime in and answer questions as they come in. So I'll go ahead and get started. I'll leave the screen up for a little bit and then I'll close it because I don't think seeing the QR code is maybe that important for too long. So I'll get started. And the first upvoted question I see is wondering if you could maybe just recap some of the key characteristics that you're using to identify different similar species within that Cariaceae family.
I'm going to put that question to Dan because I am not a plant ID expert, nor did we, I should say, we did not collect the data here, right? These are data that are collected by PI surveyors that we aggregated together. So we can't speak to the specific methods used by every single person, though they are generally broadly very similar. But I think Dan can add some nuance to that question, that answer.
Yeah, thanks, Alex. Yeah, so if you see the star-shaped baubles, little white star-shaped structures that Starry Stonewort has, that's really diagnostic, and then you can be very confident that you have Starry Stonewort. But those aren't always present, and they might not always show up on a rake head. So there are sort of subtler features that relate to the structure of the plant, what we call leaves in regular old plants are called branchlets in this group of algae. And so there are features of those branchlets that you can use to tell Starry Stonewort apart from other relatives in that family. And we also, any of you who participate in our Starry Trek event, We have materials for training people on identification. There's also some nice YouTube videos. Paul Skowinski from University of Wisconsin Extension, for example, has made a really nice short YouTube video that covers Starry Stonewort identification. But if you get a decent size amount of material on a rake head, the types of people who are doing these surveys, who are, you know, aquatic plant professionals, like Alex said, who do this all the time, can distinguish it from other species in that family quite reliably.
Thanks, Dan. All right, next question coming up. So if Starry Stonewort is found, can it all be killed? And if it's not all killed, or there's lack of control, aside over time, would part of a model be used to help explain that spread?
Maybe I'll start with the front end, give Alex some time to think about the modeling aspect of it. So unfortunately, Starry Stonewort can be quite difficult to kill, and we don't have any known cases of eradication. It really depends on the amount of Starry Stonewort that you have in a lake. If you have a large amount of Starry Stonewort, there's not really any evidence that the chemical treatment methods that are available to us, which are copper-based algicides and herbicides, can reduce the extent of the Starry Stonewort in a lake. That's in contrast to things like Eurasian water milfoil, curly leaf pondweed that we've been managing for a lot longer. There's a lot more research underlying it, and where we really can shrink the amount in a lake and be sort of chasing these little relics. However, what is really promising is if Starry Stonewort is found when it's a small infestation. And so that's why having improvements to surveillance is really practically useful. So there are cases, particularly in Minnesota, where Starry Stonewort has been found early on, where there's a small amount near an access, and hand pooling has been highly effective in those cases for keeping it contained and keeping the population small. We don't know of any eradications from that, and it requires ongoing, sustained effort multiple times per year over multiple years, but it's really effective. So the management effectiveness is highly dependent on, you know, whether you've got a little bit that's small enough that you can go out and hand pull it, or you've got a lot of it, at which point you can. The algicides can be really good for localized reduction, kind of reducing those nuisance levels. You can knock the biomass way back, but it will return.
Thank you, Dan. I doubt that I'm about to give the answer that this person is hoping for, in part because I'm not totally sure I understand the question. So they should feel free to shoot another question through if this is not satisfactory. But I think it is worth noting, right, that in any predictive model, the patterns you find depends fundamentally on the patterns you went in looking for in the first place. Like, what do you know about these spots? That limits what patterns you might be able to find. And the elephant in the room in any model like this one is management, right? We do not have actionable data that we can incorporate into a model like this one that factors in management. Like, oh, it's not there because somebody came in and killed it, right? That's a whole nother set of processes that could yield zeros we don't necessarily know about. It's not that those data aren't out there. It's like they're not interoperable with the data that we have, or there's some mystery to them. So I think there is a potential blind spot in this model, and it has to do with management. Though one might ask, are we trying to predict what is happening even in the face of management, or are we trying to figure out what would be happening pre-management in a lake? I think there's some important questions to be asked there, but it's possible that some of these lakes we included were being managed and we just don't know it, and that's influencing the patterns that we're seeing. So hopefully that at least gets at some curiosities related to management there and modeling, even if it wasn't the audience member's exact question.
Thanks, Alex. I've got another modeling question for you. So it says here, you're predicting where Starry Stonewort will occur within a lake. Is there a way that you could predict where Starry will show up on a lake level? So for example, what are the chances of XYZ lakes being infested over a certain period of time?
Yes, I'm glad you asked that question because that's a really good question. Yes, between lake risk has been modeled many times previously. In fact, if you go on AIS Explorer, our big dashboard for decision support in the AIS space, Starry Stonewort modeling for risk at the lake level is explicitly modeled there, and it's been modeled a number of times. Dan actually probably knows about this even better than me because some of these efforts predated me. But actually, what's interesting about those models is that those are easier to construct because you can use lake level variables and lake level data. If you're trying to figure out where to go look in a lake, that's harder because you need data at the level of individual spots in a lake. And that data is obviously pretty difficult to gather, right? Like chlorophyll A data can be gathered at the whole lake scale and applied to that whole lake, and lakes can be differentiated on that level. But chlorophyll A spots, like at different spots within a lake, that would be very, impractical to gather those kinds of data. So what I would say is yes, absolutely. And what is novel, or part of what is novel about this effort, is that we're not modeling at that scale because we can complement that scale. There are other models out there and other modeling efforts that can tell you, which lakes are likelier than others to have starry. This model is meant to inform, all right, you've picked your priority lakes. Where do you go look in them for starry if you can only look 10 spots, for example? I will also say, though, that I think our model could be extrapolated out to lakes you think might be infested because it'll give you an occupancy probability for any spot in a lake. So you could put into it, 50 spots from a lake and you get some occupancy probabilities back. And you could use those as relative metrics of how likely this lake is to be infested, right? If most of those probabilities are low, it's not likely that this lake has starry. Whereas if you're getting back a lake where half the spots have high probabilities, and that's a better sign that this seems like at least it would be a favorable lake for starry, whether it's there or not.
Great. All right, I'm going to get to the next question. And as soon as I ask it, I'm going to turn off my screen share, which might toggle your views. So I'm just going to give that warning so it doesn't mess you up as you start to respond. Alex, this is another one for you. And you talked a little bit about PI charter at the beginning. So one of the questions is, have you added the data from private companies that work with lake associations in identifying like Starry Stonewort or Eurasian water milfoil into PI charter?
Okay, yeah, I'll answer this at a very general level. We have PI data from everyone who has sent us PI data. That includes the state DNR, it includes counties, it includes soil and water conservation districts, it includes watershed management districts, it includes park districts, it includes a whole bunch of other groups that I'm not even sure of. Lake associations have sent us survey data. There are no restrictions or limitations on who can send us data. It is who has sent us data. And if there are private contractors out there that have data, they can send it. We'd be happy to have it. Many have. If you go to the leaderboard tab of PI Charter, you can see a fully up-to-date list of who has been sending us surveys. And we would always encourage more folks to send us surveys. So I don't know if this person was asking with a particular bent in mind, but this is all volunteer, right? We know that this data, you have to dredge it up. You may have to clean it up a little bit to be able to send it to us. We've made the submission process as easy and as seamless as we can make it. It still takes a couple of minutes per survey. So, I know that one challenge for private contractors in particular is, when am I going to have the time to submit these data? Or when am I going to be able to allocate staff hours to submitting these data? And that's partly why recently we've added to PI Charter mock contract language, where it can actually get baked into the contracts where these surveys are being done, that it's an expectation these data will be submitted on both sides, and that can be baked into the price if needed, to try to facilitate that a little bit.
This is maybe getting a little too into the weeds, but I'll add in some cases, like associations, when they hire consultants to look for AIS, the survey will be a meander survey instead of a point intercept survey, because that can be, a more cost-effective and efficient way to find new AIS. And data of that type those are not PI data, and they can't fit within the structure of PI charter.
Yes, that's a very good point. Yeah, if you think about it, meander data are exactly those kinds of data I just said earlier we can't use for this because they're biased by our preconceived notions of where we think we should be looking for a species. If we're zigzagging around a lake trying to look in specific spots we think we're likely to find a species, that's just going to confirm what we think we already know. It's not going to challenge our preconceived notions. So it's not that meander data are not helpful. They absolutely are, but they're distinct from PI data in a way that's important, which is why when Macerx started aggregating plant survey data, we started with PI data. It's not to say that we may not be interested in meander data and other kinds of survey data later, because they are themselves also quite useful. The other challenge with meander data is that they don't always catalog all taxa that we're seeing, right? They usually have a specific set of taxa they're interested in looking for, and they may not record comprehensively, whereas with a PI survey, that's the whole point, is we're trying to catalog the whole lake plant community.
All right. So Alex, thinking back to the map that you had of, I think it was Cedar Lake, there were some red dots that were near the launch. Then you had mentioned that there were some kind of up away from the launch. And one of the questions is, do you think it's possible that that's like a popular fishing area where boats commonly go and fish and might anchor in that area? So like might be dragging from that launch to a specific spot more likely?
I love these questions. These are such good questions. This one gets into hypothetical land, and I feel like a Supreme Court nominee where I'm like, I'm not going to engage in hypotheticals, but uh, but I don't know. That's something that I feel like is sort of a frustration that I feel tangibly as the modeler is I don't know these lakes. So like, there's a part of me that like often will look at a map like that and go, what is it about that spot, right? What was the factor? Because presumably that means it's a factor we probably didn't have in the model. And it's always a fascination to me, like, what would it be? Thinking about the specific mechanism, I was thinking about, okay, you have Starry Stonewort propagules attached to your boat. You're launching in the boat launch, but you're going all the way up to the north end of the lake. Wouldn't you think that a lot of that stuff would have fallen off by then, right? And thus not made it all the way over to that spot. But maybe maybe it's like breadcrumbs and Hansel and Gretel, right? Maybe bits are falling off all the way along, but they're falling into deep spots in the lake. So they're just not establishing along that route. It's not until we get over to that other spot that's maybe shallower that we have those favorable conditions for a propagule to establish. Or maybe, as you noted, it's about anchoring, right? It's about where you stop for a long time and thus can deposit a number of propagules. So it's a really keen insight. I love that line of thinking because it gets at mechanisms, right? And that's the whole goal here. We were trying to get at mechanisms too. What is it about these spots that seem to make them more likely to have starry? And if we could have included information kind of like that, I think that would have been really eye-opening. So that could, as far as I'm concerned, that could be the next study right there that you've articulated.
That's great. Always ideas to fund the next one. Okay, next question is bringing in another modeling tool, and it's wondering if findings from this project have been included in AIS Explorer updates. And I'll drop a link to AIS Explorer into chat for those who might not be familiar with it.
I love that we're talking about AIS Explorer. So yeah, AIS Explorer is the decision support tool. It basically ranks lakes by risk that they're going to become infested over the next couple of years based on modeling work that's been done about how invasives seem to be spreading around the state. It kind of gives you some indication of what would happen if you intervened at certain lakes, like can you stop infestations? Importantly, all the data and the modeling that have gone into AIS Explorer are at the between lake level. Is this lake at risk? If we intervened at this lake, what would happen? We haven't incorporated this data from this project into AIS Explorer because it asks a different level of question. It asks, if we got to a lake, what should we do there? which, if you think about it, is really an important complementary question to ask. That's not to say that it won't end up in AIS Explorer, and I do think that the next question here is how can we build a tool that would use these data? And I'm actually working on that in my official capacity right now as sort of almost like a passion project. So you'll have to stay tuned for that a little bit. But I do think there needs to be some sort of predictive or at least exploratory tool where we could use these data, and we're working on it. But it's not an AIS Explorer yet, and probably won't be because I don't know if that's the right place for it. If that answers your question.
Thanks. It's always great to hear about your newest passion projects. All right. Next one is wondering about how you defined a private launch. So they included some information here about like how busy it is or in and out boats per day. But what was your way to maybe finding and identifying and labeling them as such?
Sure. I wish Wes was here because this was many afternoons of Wes's time. So we have the DNR maintains an absolutely fantastic curated list of public launches, not just theirs, but federal, state, local, tribal. If it's a government enterprise, municipal, it's on that list. There's something like 3,600 of these across the state. But obviously, we know there are more places to launch a boat at a lake than just that. So what Wes did is he got on Google Maps on satellite view, and he just went meticulously all the way around the outside of the lake and said, does this look like a thing you could launch a boat at? And then he'd say, what does Google say is near here? Right? Like, is there a bar here? Is there a bed and breakfast here? Is there a marina here? Is there a boat club here? Some cases we didn't even know what it was. If it wasn't on the DNR's list, but it clearly looked like it was a boat launch that you could launch a motorized boat at, we counted that as an access, but labeled it as private. So it's kind of this grab bag of accesses that clearly exist, but we don't really know their story. And that's the challenge, right? Is we don't know all these lakes firsthand, and we can't always know much about these lakes firsthand. So what are all these accesses? I'm not sure. And we would have loved to explore, deeper there. Like, okay, are there sub differences between different kinds of private accesses? And we didn't get into that. So private, public, even those terms are fraught. They're kind of our internal jargon for these things, but I don't even know that those are really the right words. I just don't have better ones. There are things that are not on the DNR's list. Let's put it that way.
And I would say generally they're commercial, multi-user public accesses. So they're resorts and marinas and those types of things.
A number of pubs, a surprising number of bars and restaurants have them, which kind of blew my mind. That is not something I think of as having a boat launch, but maybe it's just because I'm not a native Minnesotan, so I don't realize how seriously you guys take your lake access.
And we did include Wisconsin, so that's going to help in terms of bars.
True. Okay, yes, right. Good. Thank you for pointing that out, Dan.
Great. All right. Next question is, do you plan to run this model for other invasive species?
We have joked / dreamed about this because Starry isn't at that many lakes. And I said earlier, right, if you're going to look for reliable patterns, you need a giant data set. And this is a giant data set relative to other data sets, especially for Starry. It's a huge data set. There are something like what, Dan, 500 lakes in Minnesota that have Eurasian water milfoil? Just in Minnesota. So does PI Charter probably have a giant data set already that could be leveraged on something, say, like Eurasian water milfoil or curly leaf pondweed? Absolutely. Could we get at more nuance here if we looked at that, like another species that had that large of a data set? Absolutely. One limitation there is who's going to sit at the computer and look for all these private boat launches across 300, 400, 500 lakes? It would be a thing. And honestly, if there's been any deterrent at all, it's been that is just the level of like data assembly here might be kind of daunting. But keep whispering about it and maybe it will happen because it does interest me.
That's great. Thank you, Alex. Okay, it is 1:59, so I'm going to share my screen one more time while we wrap up. Thank you to everyone who joined us today for our webinar. Thank you, Alex, for coming to present, and Dan for being here as well to help out on the Q&A panel. The couple of final housekeeping items before we tune out is to watch for the recording on our YouTube channel. If you registered, you'll get a note when it's live on YouTube. Again, we have to transcribe it, so give us a week or so and it'll be up for you to view. You can share with others who are interested, go back and review something that you wanted to listen to in more detail. And you'll be getting a survey as well. I think it pops up when the window closes. You might get an email, but we appreciate you taking some time to fill out the survey after each webinar. Finally, there's a couple of emails up. There are questions that were asked that we did not have time for. So if you really wanted an answer, you can bug Alex. His email is showing there. So feel free to shoot him an email. And if you have questions kind of more generally about like the webinar or AIS Detectors program, our email address is showing as well, and you can send a message to us there. That is all that we have for today. So thank you for joining us. And we'll look forward to hopefully seeing you at the next webinar in 2 months. You can watch your inboxes for registration reminders about that one if you haven't already.